International Women's Day.
Hey guys, so we got Keisha from BioAmendments coming up. I definitely put her as one of my favorite people for sure. I feel like her and Casey are definitely doing the Lord's work. They're taking Elaine and her work, her years of work, and just teaching it to everybody. And that's one of the most important things you can do is educate. So a huge fan of Keisha, Casey, and BioAmendments. They bailed me out. I used to make my own compost for years. I used cannabis to make my own compost. And then about 2 years ago, the state told me I could no longer do that. And you guys know how big of an input compost is for the living system. So it's hard to find, Elaine Ingham style quality compost. It's not readily available at every hardware store, So having people like Casey and Keisha that are willing to put in the time to do it right, to make sure that when we need it, we can have something like that is truly valuable, guys. I hope you guys can appreciate it. By them making good compost, to make our job easier. Trust me, that's the bread and butter of it all. So I appreciate you guys. Thank you guys. Let's rock this thing, Keisha.
Adjust here. I think I like that. All right, we got people coming in, so I'll just get started talking about myself a little bit. My name is Keisha Ernst. I am one of the owners at BioAmendments. Hopefully, a bunch of you got to come and visit with us yesterday during our compost class, where Casey and Danny and I were teaching you how to make your own microbes. I shared then that I'm an accidental compost producer. I didn't get into this work to make compost. I got into this work to be a consultant. And like you've heard so many times, it's hard to find good compost. Especially compost that when you put it under a microscope, you're going to see all the microorganisms present: fungi, bacteria, protozoa, nematodes. We even saw a microarthropod today on someone's sample. I don't know if you guys got to see that, but it was super cool on the screen in there. So when we were thinking about this talk, I was actually feeling quite uninspired to come up with a name. I knew that we wanted to bring the microscope out. I knew we wanted to get the soil up on the screen. We wanted you guys to be able to bring in your own soil, your own compost, and just see where you're at. Just a baseline of what life is in that. We also knew that we wanted to teach you how to make compost. So our compost is actually best used in the soil with plants, or to be grown into a larger compost pile with more microorganisms. We like to think of ourselves as a composter's composter. We like to make inoculum for people that are doing this work so that they can grow it out on their own. But when it came time for this talk, I was imagining that I was going to teach you guys about how to apply compost extract, how to apply compost tea. It was going to be a very specific talk on how to use this compost best at home. And then I started thinking about it, and actually everybody that got a ticket to this workshop actually got a recording of the talks that we did last year. And Casey and I spent an hour going deeply into sprayers, extractors, different ways to use this stuff. So I recommend watching that talk, because I've totally flipped what I'm going to do today. We're not going to go into application. We will tomorrow. We have another workshop tomorrow. We're going to make a compost pile, and we're going to talk about how to use microbes. We're going to do some live demos with extract, and just get more in depth on that. Now I have a compost company that's running. You guys have met Danny. He's up at the booth most of the days running the microscope. He's the one on the ground now who takes care of our compost. A lot of people think that we're just building windrows all year long. We're not. We make just enough compost for our clients, our big farmers that need it every year. We get an idea of what they're going to purchase, and we only build once or twice a year, because most of the time now we're consulting. There's a huge shift in our market. In the beginning, you could imagine it was almost all cannabis growers that bought our compost. It was expensive, and really farmers at large weren't using microbes the way the cannabis was. So there was a point about 2 years ago where it started to really shift. And we got away from selling to so many cannabis people just because the industry was going down. But farmers really took to it. So we started working a lot with walnut farmers, olive farmers, people producing turf, hay, And they really took our understanding of these processes to another level. So Dr. Ling was my teacher. She gave me the foundation for everything I know. She taught me how to use the microscope, how to make the compost. And I have to say, I used to go hard on the compost. I would put a lot of that material into a small space. But it was my farmers actually that taught me that less can be more. Excuse me, I'm a little dry. We work with a guy named Brian Vaugh. If you guys have ever watched the Soil Food Web School lectures, you'll see him all the time kind of overseeing our talks. And he is really, really into the data collection. And so when we started our compost lot, he started working with us, and we were kind of in tandem doing these huge consultation projects. And he's working with these farmers that are investing heavily in biological analysis, chemistry analysis, plant sap analysis, and also tissue tests, right? So these farmers have been following their nutrients and what their plants are taking up through the years. They're using this data along with the biological analysis to show, hey, are these microbes that we're seeing under the microscope actually providing the nutrients that my plants need? And when we use all of these different metrics, when we use them all together, we can see a fairly clear picture of what our plants are able to uptake and what's missing in the system. Right? So we're not just using microorganisms. We've graduated to learning more about transitions. And so what we found working with these large-scale farmers is sometimes we're only using this compost at the rate of 2 pounds per acre, which might sound kind of crazy and like hard to believe, like, you know, what is that going to change? But you know, farmers, big, large-scale farmers, they're all about the bottom line. They're very good at knowing how much their processes cost, and how much money they're making per acre. And so it was actually them that taught us that you know, our compost isn't super expensive compared to chemicals. It's actually, the trends we're seeing is in the beginning, we're a little bit more intensive. They're applying frequently, but they're applying very lightly. And we are seeing, you know, just amazing results from that. All right. And so when I started thinking about this talk, it was like, I was thinking about money, like microbial wealth, like what's the actual profit that we're going to get from working with microbes. And so I'm not going to go too in depth. I'm going to keep this presentation fairly simple. I think Matthew Bates really showed everyone clearly that this is a complicated world when we're talking about microbes, when we're talking about plant interactions and the symbiosis between these communities of organisms. Like, it can get just mind-bogglingly complicated. But what I love is what Elaine taught me is just the simplicity of it. I feel a lot of the times the fact that it's so complicated, it gets in people's way, and they don't really want to. They don't want to get started with it. It just feels like too much. So, to be simple, let's just talk about fungi and bacteria. How can they make us money? You know, how can they bring value to our life? Well, fungi and bacteria are very specialized. They have enzymes that can eat rocks. Nobody else in this room, nobody else on this planet can eat a rock and pull nutrient from it, right? They are very specialized. When they're doing that, they're creating these glues, and they're binding, they're pulling. Take a look at the microscope. When we have the microscope up, come over, ask us to point this out to you. I can show you in a soil sample where these bacteria and fungi are reaching out, grabbing and binding together these inorganic and organic materials and clustering them into these aggregates, right? That's what creates soil structure. So what comes from soil structure? Our roots can grow deeper, we can access more nutrients, we can store more water. How do you actually get the metrics on how much money you're making from this? Our large-scale farmers do, in a way. They're actually tracking the organic material and the porosity of their soil over the years, and they're showing, wow, when it rains now, this microbe sponge, this soil sponge is holding my water. I don't have to be out irrigating. I don't have to worry about it. It's a stress, you know, it brings the stress away, and it actually does save money, not having to pay for the water or for the people out there on the fields worrying about it. It also cleans up toxins, and that's in all aspects of our environment, in the air, in the water, in the soil. Fungi can just bind to these things. It's like incredibly complex. And so, what is the value of being able to clean up a toxin? You know, a lot of this value comes from things that just aren't bad that are happening, right? It's like, you hear this, the solution to pollution is dilution. The only reason that that statement exists is because of microorganisms. Now when we go out into the environment and we start killing them, we start, basically eradicating them from our fields, what's going to clean up the pollution? We can't dilute it and fix it anymore, it's just going to start stacking. So these guys are very specialized in eating the things that we produce that are making us sick, that are hurting the earth, right? So there's a lot of wealth behind these organisms that isn't just financial, but you can start to see how there are huge financial gains to this. If you're talking about heavy metal, toxic waste cleanup, my goodness, being able to make soil useful to grow plants in again, that's a big deal. Right, then there's also protozoa. They do not get enough attention in the media. You hear about fungi and bacteria all the time. There's bacterial powders, there's fungal inoculants, everyone wants fungal diversity, you hear that all the time. Or fungal domination, but really what we like to think about is diversity, right? We want a lot of different species of fungi and bacteria, but we also want stuff out there eating them. If we don't have the microorganisms in our system to eat the bacteria and fungi, which are basically they're just packets of nutrients sitting in the soil, doing their good works, all their great side effects are happening, but that nutrient isn't getting released until something eats it. And that's where we look at protozoa. These guys are like the wolves of the compost pile or the soil. They are cruising around. When you see them on my screen, they're all just kind of chilling, sitting, not really moving anywhere. But they can boogie around. When they actually have these pseudopods, they can reach out and grab bacteria, pull it in and eat it. So when these guys eat and poop, they're basically making plant-available nutrients for our plants, like right there in that moment. And I get into nematodes. Nematodes are awesome, everyone loves nematodes. They're the most popular. You can see them really easily. Lots of different shapes and sizes, and they're always moving around. So that's always the star of the show, and we're on the microscope. People want to see those nematodes. But often, it's just the bad guys that get the press, right? And it's again, good, bad. Root-feeding nematodes are not evil, just like deer are not evil. But if you know what it's like if you live in an area that has an abundance of deer, they'll come in and just smash the vegetation if they don't have predators. If they do have predators, then they're helpful. They help us deal with our green materials out in the forest and our pastures, and it's not a problem. So with the nematodes, it's you're going to want to find as much diversity as possible, and you've got to think about what their role is. They're not just pooping and creating nutrients. This is a biocontrol. So a lot of these pests that Matthew was talking about earlier today are these nematodes' favorite snack. So these guys they're our partners. We can work with them. I think of them as my coworkers. I think of them as my boss. If you want to talk about microbial wealth, every bit of my money comes from these microbes. And so I spend my time on a microscope looking at them and figuring out what I need to do to help them, further their existence. All these benefits that we get from these microorganisms, the microbes aren't out there I'm going to save the soil and protect the planet and help you digest food, right? That's not it. It's just a side effect of what they do. They're out in the world just being themselves. They're just operating in the way that a microbe operates. They're eating, they're pooping, they're replicating, and they're getting eaten by something else. And so in a really, really simple way, as long as we make sure that there's a lot of microorganisms in one place, they're going to do their thing. They're going to eat each other. And they are, like Matthew said, they are going to learn how to not be eaten. So over time, pests, pathogens, and beneficials are all operating a little bit differently just to be able to stay alive longer. All right. And so what about the benefits from this that, money can't buy? And we can look at reduced fertilizer costs. That's huge. If you look at fertilizer costs, have over doubled in the last year. And we're talking about large-scale agriculture. By just reducing those numbers, the number could be millions. Some of the farms we work with are saving millions of dollars every year, basically only because they're reducing their input costs. Right? But then think about the other things. So your plant system is functioning. You have a healthy soil food web. Anyone out here that has started working with microbes kind of knows what I'm talking about. You're going to have these things that you notice. It's not perfect right away. But maybe all of a sudden you don't see as much powder mildew. Or maybe the plants are just a different color green. Maybe your root systems are bigger. When you pull your plants out of the ground, you're like, whoa, that's different. These benefits you're going to get, they're going to stack. They don't come all at once. This isn't a buy our compost, put it on your plants, and you're set for life. That's really not it. The idea is to actually change the way you think, to be focusing on the microorganisms, right? So whenever I think about what I really get from this, like what is like the best thing about working with microbes, working for microbes, it's my soil community. It's you guys. It's being able to come to places like this and be able to share what we learn and share from other people and learn from other people. We can reach out like fungi, we can operate like microbes, and we can be the best people that we are. We come into this community, right, and I look around the room and there's a lot of different people here. You guys all have different functions. Just like when I look under the microscope, right? I don't know exactly what your passion is, or like what your favorite food is, or what your mom's name is, but if I spent enough time with you, asking prying questions, paying attention, I'm gonna start to gain that information. It's a lot like that with the microbes, right? Like I look at them under the microscope and I don't know what this testated amoeba specifically is gonna do for my plant, or how much money that fungi is gonna make me, but I do know that if I'm focusing on these microorganisms, I'm focusing on soil, I'm doing my best, and getting to come and do these sort of things, we are actually like the microbes. Now we're coming together, we're doing our best, we're just living our lives, and the side effect is actually pretty brilliant. We're all making connections that are strengthening us, we're all making connections that are gonna make our work more impactful, did a weird thing with the E's and the S's. Anyway. It's the experiences, right? Like this is the stuff that really makes the juice worth the squeeze. It's a reflection to me. I often think about, when I'm thinking about the soil microbiome, I think about it the way that I think about people. We were talking about good microbes, bad microbes, who's going to express themselves in a terrible way. I think it's like that with people too. If we have a mission and we have a passion in life and we are doing our thing and we are keeping our company diverse and learning from these people, it's just like this whole fruit. It just makes everything grow better. All right, so I was thinking about, like, as far as application goes and how we think about all this, what are the few things that I could say that are actually like things that get in the way of our clients? The number one issue that the people that we work with have is just not getting started, not trying. They'll invest in the soil food web school, they'll buy a microscope, they will literally go and pay hundreds of dollars for consulting hours, thousands of dollars for compost, and just never take that first step to get started. And it is a lot of work to be constantly thinking about a force that's invisible. It's not easy to wrap your head around working with something that you can't see. And so, that's why I say, like, the microscope is really your biggest tool when we're talking about working with soil microbes. We want to always be thinking about what's hurting the microbes, what's helping the microbes, or what isn't affecting them at all. So in the most basic way, when we work with our clients, we're going to go through and we're going to find out what is your standard operating procedure. How do you treat your crops? And that's your first thing to explore. Maybe you don't have an official SOP written out yet. Maybe it's just in your head. But you can, when we started writing our standard operating procedures down, it really started to solidify what we were doing. And we were able to look through each thing. Tilling, is that going to harm my microorganisms? Yeah, it is. Do I need to do it to put my crop in the ground? In some cases, yes, you do. And so it's not always saying, well, I'm not going to do anything else now that's going to harm microorganisms. No. I'm going to do my best not to harm microorganisms with what I have. So when we're talking about large industry, when we're talking about, especially like fruit and nut trees, these guys have multi-million-dollar machines that harvest this fruit, right? And so a lot of the times they have to have procedures that are going to be damaging microorganisms. And when we define those things and we say, all right, you guys are getting ready to put sterilants out under your trees, what's that going to do? It's going to sterilize the soil. It's going to kill almost all the fungi, all of our predatory organisms around the trees. Well, now what we do is we adapt that procedure to say, okay, after the machines are out of there, after we're done, we're going to come back through with a compost extract, with some solid product, who knows, whatever you have access to, and you're going to put it on the ground. And so it's this 3 steps forward, 2 steps back reality, where it's like you're doing your best to think about it, and then trying to, get in the way of it and bring more of the life back, right? This happens all the time with fungicides. We'll have a large-scale farmer that, they've been working hard on getting the biology out, and it just isn't quite doing it. They have something come up where it's like, I'm going to lose this whole crop, or I'm going to use this fungicide. Well, they're going to go ahead and choose the fungicide for sure, but what we're going to do is step in after that and say, okay, hey, can we mitigate that damage that was just done and try to get some more microorganisms in place? Now there's something about the continuing to do this over time. You can imagine like if someone just took us and dropped us in the desert. Right now we have no food resources. Matt Gates talks about this a little bit. You want to prime the environment for the organisms that you're putting there. So when you're first getting started, even if your compost extract is just ridiculously full of diverse organisms, like who's going to be able to live, who's going to stick around, we really don't know. And it's not unless, like when we're using the microscope, we get a good idea. It's still not perfect. But if you think about it, those microbes that land, the ones that take hold and they start doing their good work, right? The fungi starts to bind things up. The bacteria starts to stick particles together. Some protists start to eat, right? A cycle is starting, and it's starting to create a real, true, porous soil. Well, now when you come back again a few weeks later or a few months later, depending on your resources and what kind of time you have, and you seed in microbes again, well, you might catch some more this time. It's not an exact science. It's more just the art of continuing to try. What else was I going to say on that? Yeah, I would say too, like, so with the microscope, you're only getting a really above view, right? You don't know precisely what's going on. We can actually use DNA analysis now. There's a company out of Sacramento called BioAmendments, I believe. It is very, very interesting to get tests with these people because it'll start to show you what organisms are taking hold in your soil and what they actually eat. And we've seen a lot, like the bacteria and the fungi that are taking hold in the soil far before we see the predatory organisms wake up and start to be active are salt-tolerant, salt-loving microorganisms. And so, it might take a while for those earliest, those early like pioneer microbes to start to do their work and to start to really create a good soil system. But as time goes on, those more sensitive microorganisms are going to be able to take place. So the other thing is just a refocus, like I was saying before, refocus completely on microbes. It's not the only thing you have to think about. I mean, I'm kind of that way where I'm very suited to think about microbes all the time. But I think most people go through their day-to-day just not really understanding that these creatures are there. I definitely see it on the faces of people that are looking at the microscope for the first time, like, whoa, like, there's stuff in there. Like, they are alive, they are moving. I mean, it almost makes you question veganism. Like, there's life everywhere. All over this planet, there are these little creatures that are living and eating and being eaten. Right? So that refocus of just, and even if it's something small, I'm not going to get into the very specifics of how you're going to cultivate the best microbial habitat, but you can get on the internet and just look at permaculture for that, right? Like, we all know that we need to cover our soil. We all know that bare ground is not going to be a great host for microorganisms. Now, we could level that up a little bit and say we do know that microorganisms exist with plants. They've been working with them for years. And so something a lot of people miss, something that kind of just goes over the head, is that it's not just your growth season. Like when your cash crop is in the ground, that is not the only time to think about your soil. And this is where you gotta get a little bit more microbially minded and think, what does this look like over the whole year? Do my standard operating procedures stop in September or October? Or do they go through all year long? Like, are we thinking in the fall about getting those microbes out? That's the best time to get started. And for me, that's usually when I'm starting to wind down and get tired. I've been working hard, I've been in the garden all year, I've been using my body, and I'm ready to just chill. But that's not the best thing to do. I like to assess in the fall and the spring. Go out to my garden. I don't grow a big cash crop. I spend most of my time teaching these days. But I do have a garden, and my goal with my garden, right, this is something to think about as well. What's your goal with the whole project? My goal with my garden is to be able to go outside whenever I'm home and cut a vase of flowers for my house. It's super simple, but I can tell you when I first started living at that house, I could not achieve that goal in any way, shape, or form. Not one thing would grow in that yard except for bindweed. And they have gorgeous flowers, but they don't hold well in a vase, and they're very seasonal, so it didn't really work out for me. Now, as time has gone on, I've put a lot of energy into that. Especially in the beginning, I was putting a lot of compost, a lot of extract. I was paying attention all the time. Now my garden has flowers in it, and there's snow on the ground back home. I swear, you guys, like my flowers just came right up to the snow. Didn't have to do anything. So I do a lot less now, but every fall and every spring I do a little check-in. I look at my garden and I say, who's died, who's living, do I have any bare spots that I can address right now? Mulch is mulch is number one, but it's living mulch really, right? So the more diversity of plants that we can get into the ground with our microbes, that's the best way to store them, that's the best way to grow them. A lot of people ask us, how do I best store your compost? How am I gonna keep it alive for the longest amount of time? Well, first of all, water. That's always important. But secondly, our compost doesn't want to live in a plastic bag. It wants to be out in its community, working with plants in the soil, growing. So the number one best thing to do with your compost at the end of the year when it's fall, get it out on the garden. Get it in the root zones of the plants that are still in the ground. Like, get it outside so that it can continue its life. And then just make more compost in the spring. I think a lot of people, they get the compost, and you don't have to buy it from us to feel this like, it's my treasure. Gosh, you make it yourself. You put so much energy into this yard or two of compost, you don't want to let it go. And it's a barrier for people. It's a barrier for me. When we start to run out of compost, I start to get real stingy and weird with it. Just gotta remember that, like, you can grow more. So Steve had this crazy, crazy idea. And he was like, you know what, everybody that's here has learned from Dr. Elaine. I'm sure, can we get a show of hands of people in here who know who Dr. Elaine is? Where she's influenced your life in one way or another? My goodness, Elaine. Take a look at that. This woman has been working on soil microbiology longer than some of us have
Elaine, Izzy was talking earlier about when we get microbes starting early, the salt bacteria kind of start. So it got me thinking about succession evolution. In terrestrial plants, we get clearly an evolution from prairie out to forest. Does that happen when we start with kind of maybe dead soils, moving into richer biomes? Do we see a succession of microbial development? And do some of the bio introductions, the biostimulants, actually cause problems with that evolution of microbials because it's interrupting or it's introducing something in the wrong succession?
It varies a lot on whose side you're looking at this. Because if we're human beings and we want to put in orchard trees, we know that we've got to get more and more and more and more fungal, starting from strictly bacteria. That's what, we have to start with. Those sugars are going to grow bacteria, but that's not the plants we want. Those are weeds. Those are the early successional things, the grasses, the pretty, but may get in the way of a good production. So from that point of view, we're going to go in and we're going to make certain to move that successional relationship between bacteria and fungi, so that we move as rapidly as possible to orchard systems. So what's the fungal to bacterial biomass ratio for those particular plant species? It should be at least 5 fungal, 5. This is what B71 does to you. So I made it. I just can never let go of talking about my favorite organisms on this planet. So fungal to bacterial biomass ratio for the tree systems is going to be up there in the 5 fungi for every 1 bacterium, up to maybe 10. Well, in some cases it might even get up to 20, 25, somewhere in that range. So you're going to be doing things, if you're at that first stage of succession, where what is that fungal to bacterial biomass ratio for those plants at that stage of succession? It's like 0.05 fungi for every 1 bacterium. So you're going to be there's going to be very different things going on in that soil. when we're at that stage of succession. Get a little bit more fungal, and now you're going to be growing herbs, you're going to be growing lettuce, you're going to be growing things that have a fungal to bacterial biomass ratio a little bit more on the fungal side. Keep going every stage along the way till you get to old growth forests. So what is the fungal to bacterial biomass ratio of some of the trees who are more than 2,000 years old? We've seen as much as some things like 5,000 parts of fungi, micrograms of biomass of fungi, as compared to one bacterial. Strongly fungal. Yeah, but then flying squirrels, for example, in
Oregon Redwood Forest.
Oregon Redwood Forest, thank you. I was trying to think of the name of the state, Washington. And it just takes a little slower time to access facts from my mind anymore. So the flying squirrels, what do the flying squirrels mostly eat? Fungi. So you may start with, well, the mammals in the system are sound asleep. They're haven't finished their winter relaxation, and when you try to get your hands into that soil, you can't get in there and pull anything out because the weave of fungi is so intense that you can't pull it out. You're gonna have to go get a really sharp knife or something with a really pointed edge to pull out a handful of that soil, because it's just interspersed so completely clogged up. From our point of view, it looks like, oh, there's going to be a problem with infiltration of the water because we just have so much fungal biomass in there. Okay, so interesting. You now come back after the snow's melted and after the squirrels have woken up. And you go looking for fungi in the soil and you almost can't find any. They're there, but you've got a wonderful structure on that soil. You're getting good infiltration into the soil, but those mammals have eaten a huge quantity of the fungal biomass in that soil. Okay, so as it goes through the summertime period, we're going to have less and less fungal biomass clearly sitting around. We're back to the single strands that you can't see with your eyeballs. So, oh no, our fungal to bacterial biomass ratio is now all messed up. Wait until it starts to get cold. And all the mammals, the flying squirrels, find a nice burrow up in a tree someplace and go to sleep. What starts happening in your soil? You've never seen fungi grow that fast. It doesn't take more than a couple weeks, and that soil is starting to feel just like that initial sample that we took, trying to get our hands into that soil. So, just massive amounts of fungal biomass. So, you got to think about these things when you're working on them. What do you want? Is it okay that we go out and destroy those first few early successional stage successional places? Yeah. So, ethical problems exist, don't they? I kind of feel like it's an imperative of nature that those pastures or large areas are going to move along into a higher and higher state of succession. But the highness about that is just seen from the human point of view. Mother Nature seems to have that same point of view. because she's gonna really push to get to that old growth situation. Never quite gets there in a lot of instances. So is that good or bad? What did we want growing in this acre of soil? Do we have the right to make those kinds of decisions?
While you're on succession, would you mind talking a little bit about nematode succession? Because I remember when we first started getting fungal feeders in our compost pile, and I called you so excited, you were actually explaining to me, how quickly these organisms reproduce and the actual successional reality around nematodes. Will you speak to that a little bit?
Well, it's a predator. The nematodes are bacterial feeding nematodes eat who?
Bacteria.
Very good. Fungal feeding nematodes eat. Omnivores eat. Yeah, whatever that goes in the mouth. So, and then root feeding nematodes, what do they eat? And they have very strong preferences for the part of succession that we're looking at here. The root feeding nematodes are going to go after a pore, an area where we don't have the concentration to let the root feeding, not to wipe out the root feeding nematodes. That's what I'm trying to say. So one of the things that occurs as you move through succession is that you have a greater and greater and greater ability to stop the pests, to stop the diseases and those organisms that we don't want to have in there. So that the bad guys are left in the lurch. Well, are all root-feeding nematodes of equal badness? And of course, yeah, really they are, because what if those root-feeding nematodes are eating your weeds? Yay, go nematodes! What if the root-feeding nematodes are eating your sequoia seedlings? Kill them. All relative, isn't it? It's kind of fun to start thinking through all of these. Are all of the nematodes good? Is that the kind of answer?
I also would like to, I was surprised at how quickly they reproduce. So how many young typical bacterial feeder, and then when you get up to predatory nematodes, how slowly they reproduce, the numbers you gave me were astonishing. And it helped me to think why it's so hard to build these numbers up once we destroy our nematodes, right?
Well, I'm not sure that I'm going on the same wavelength as you are today.
Well, what she'd been mentioning to me was that bacterial feeders can reproduce really quickly. And so a lot of the times we'll see in our compost these booms of bacterial feeders. I've had some people talk to us today about it where they're I have 300 bacterial feeding nematodes per sample or per drop. And it's because they're reproducing super fast, right? And we're not often seeing fungal feeders, but they're more slow. They're not having their young in mass like bacterial feeders are, right?
Right.
And then you told me predatory nematodes. Some predatory nematodes can only have one baby per year. They just don't reproduce fast. They don't have a litter. They're not putting off hundreds of eggs. They're just and they might take years to reach sexual maturity as well, right? So they're almost like people in how long it takes for them to actually reproduce. And so think about, we go out and we crush a field somewhere, and we've knocked out these basically nematodes that can only have one baby per year. How long is it going to take to get them back? Yes.
Well, is that in correlation to the reproductive or generational series on, or what they're feeding on? Bacteria reproduces very quickly.
Right.
So obviously a predatory nematode should reproduce very quickly as well, whereas the other ones, the prey, reproduces at a slower rate. Therefore, we don't want the predator to reproduce as
As
quickly.
quickly, yeah. I mean, it makes sense, right? I mean, when you look at larger organisms, when you're talking about predators, they're not having babies as quickly as their prey. That would make sense, but I don't know. That was something that I was just like, mind blown. So.
The root-feeding nematodes that are outside of the root system, they don't ever enter your root system, tend to be a lot more like fungal-feeding nematodes. They, the females will lay a lot of eggs. They may or may not all hatch, but you know, you can have quite a few of the ecto root-feeding nematodes. But when you get to the root-feeding nematodes that go inside the root system and have the babies inside the root, there's just not a whole lot of space to produce 100 nematodes of that variety. So they may only have 10 to 20 throughout the whole life cycle of that female. Yeah. So interesting. Yeah. So it is, but you know, because the root-feeding nematode that's inside the root, endoparasitic, is almost all of her babies will survive. And the babies hatch. Maybe the eggs will be outside the body of the mother, or the mother keeps the eggs inside. They will hatch when the babies are inside her body, and it kills the mom. And then the 10 or 12 root-feeding nematodes will leave that root system, and for the next 4 to 5, where you go from stage 1 to stage 2, stage 2 to 3. So that process was all done outside the root. They don't come back into that root system until those nematodes are going to lay eggs. And so the yummy things inside of the root are perfect for those nematodes to move inside that plant root and have almost all of those eggs hatch and turn into baby nematodes. So another question?
Yes, you spoke of the impermeable surfaces that you seemingly encountered in the forest, and it made me think of the detriment that's being caused in a lot of our large-scale agriculture and, unfortunately, some of our small-scale grows that are going on with multiple factors, including mainly salts. Can you kind of speak on how salts are binding up our soils and causing, I guess, calcification on the surface and binding? You get where I'm kind of going with it, how the salts bind up
Yeah, we
our ability to take water, the soil's ability to take water.
Yeah, because like if you're going to use calcium carbonate, it's a salt. If you add calcium carbonate to the soil, it's going to be pulling in a lot of the water, and that water may have proteins, sugars, various different things inside of it. Usually, then that material will have come on, brain.
Okay, take your time.
So starts with an E. So, sorry, I'm losing the question altogether. Sorry. Yeah. 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 Exchange sites. That's what I. Yeah. So E was the first letter. So the exchange sites are now going to be filled with that salt, high salt level. Yes, now with water, and held on the surfaces of the organic matter that's present in the soil, and also on the surfaces of some of the protozoa, nematodes, and arthropods. Thanks. So as that accumulates, those salts accumulate, it's going to be harder and harder to have the water in that amount of soil available to the plant. So, what do you do to get the salt off those exchange sites? You need to be putting in either acidic pH or alkaline pH. It depends on what you want to do. So typically the calcium carbonate is going to be pulled off of that exchange site by the microorganisms. At the other end of it, we have mostly bacteria, and the bacteria is typically anaerobic. So that water, that energy, the nutrients in there are generally lost. We're not going to be able to get at them until some serious changes in pH in your whatever, your soil or your compost that you're building. Lots we don't know yet. Much things to be learned.
Elaine, I was just wondering if you have any insight into composting with materials that are potentially infected with viroids or viruses. And it's something we've been encountering a lot more in the industry, and are creating a lot of waste products with the green material that is infected. But wondering about transmission, I guess, through compost of viroids.
Yeah, I would tend to encourage you to get your temperatures up to a really high level, so you would take out those virus if you can. The virus don't survive everything, so how do you know what that virus level is? You would probably want to look at your sample using a really good microscope, because you want to be discerning whether we've got dead and dying parts of the plant, are those patterns more appropriate to the virus as opposed to a bacterial feeder or something like that. So I think that's going to be the wave of the future. I think the staining that you did, where are you? The person who talked about the booth. Matthew, there you go. Not here? Because I was going to ask him what was the fluorescent stains that he used in that picture where you saw those virus very clearly, because they are stained with UV fluorescence, and you can more easily perhaps count the number of virus particles. So possibilities in that direction.
Are you aware of viroids being any more difficult or having a different heat tolerance than your average pathogen?
I would expect that they're going to be the same or very similar. No evidence to suggest that they are capable of surviving higher temperatures. That's, I'm pure guessing at this one. Let's go out and let's get the research done. How many more am I going to do?
We have 5 more minutes technically.
4 more minutes?
4.
4? So, anyone with a burning question?
I just curious on Hi, Dr. Elaine. I'm a desert grower. I think probably one of the main people I hear that leads this example when it comes to farming organically and doing the right things, probably Steve. You still see a lot of tilling going around in this town. I think back in the days, I can't remember if it was in the 1930s, Steve, or what was going on with the cotton farming out here. But from what I hear from the locals and stuff, they said it really ruined a lot of the land. They were dropping the pesticides and synthetic nutrients and stuff like that. What just basic advice could you give to people that live in the Southwest? Because since you're here, because this is what's really important to me, you know, and I think it's really important to people like Steve too. How can we utilize our indigenous microbes more? You know what I mean? Is it harmful to, or I've been really curious about bringing microbials from, you know, ferments and different fertilizers and stuff from different places and then bring them here into our garden.
Yeah,
I've been thinking deeply about that. I'm just curious, what can we do to kind of get back to the resources that we have here?
Yeah, you want to stop tilling.
Yeah,
Number
for sure.
one, the first thing to do is, you know, maybe for the last time ever, you're going to do some tillage just to make sure that that soil is broken up. You know, if you get your penetrometer and push it through that soil, and you know that penetrometer can't go more than maybe 5 to 10 inches, you're in trouble if you don't get in and break up that the compacted material. So you know that. So the last time ever you're gonna till. If you have a nice deep soil that it goes down, you know, several feet, don't till, because you're not going to be reaching down that far. You're having fun killing all of your organisms up here on the surface. So, what's the next one? Yeah, so you're in a desert system, so you want to get coverage of the surface of your soil as rapidly as possible. That typically means we want to go for a perennial, a short, low-growing plant. Pardon?
Native perennials.
Native, yes, yeah, good for, thank you for making sure I pointed that one out. Grow, you know, get the seeds of something that is supposed to be here. That if you go out and go hiking, will you see it in the higher level of the bacteria and fungi in that soil? You might have to take bring some samples home to figure out where, if those plants that are growing there, are they fungal feeders? Are they bacterial, fungal biomass more higher? Is the fungal biomass higher than the bacteria or not? So go and collect seed from all of those plants that are growing in areas that are still using the indigenous organisms were not destroyed by what we human beings have done to that land. I mean, go back in time and we used to drive cattle across these landscapes. Would you drive a bunch of cattle across these landscapes as they stand right now? Not like your cows would make it. We really did a job on all these, semi-arid, droughty systems that used to promote the growth of trees. The trees being the bushes and the trees, the shrubs, were here, and we came in and we destroyed them. I've done some work with Land Bureau folks looking at what's on top of the mesas and the places that you can't get animals up there. And you've got all kinds of juniper, you've got different kinds of shrubs, you've got trees even growing in those areas. So go collect those seeds of those indigenous organisms and use those to inoculate your soil. So you're getting both those organisms by picking up soil. You want to mix that into your into your soil, and shrubs, even grasses. The perennial grasses are quite often indigenous, and You can go out and pick the seeds out of the plant come late fall. So that's the sort of thing I would do. Trying to be as pushing that system along as rapidly as you possibly could. Now, would you utilize humic acid or fulvic acid or any of those other very hard to, they're just tough, they require a wide fungus to bacterial ratio. So which one would you use in preference? Which one promotes the growth of the fungi more than it promotes the growth of the bacteria? We're trying to go which directions in succession versus the weeds and things if we're pushing the bacteria. We're definitely going to want to be using a food resource like humic acids. They're going to be the most complex. They're going to be the most difficult to decompose. The second would be folic acids. They're a little less complex. They're not quite as dark in color as you would see with the humic acids. Ulmic acids, actually clear in color, but it is still compared to sugars and materials that we have at the early stages of succession. Even ulmic acids are going to be better than putting in real sugar. Can I add? Yes.
Right, I really want to say something to your question because we get this all the time, and it's really the number one reason that we are teaching people how to do this on their own is if you're in a very specialized area like the desert, you maybe feel called to really work with the microorganisms that are here. You could always buy a product like ours and use it. That's amazing, but you know, a lot of what we do is to preserve these microorganisms. You know, whenever whales were in decline, people did not know that there were whales in the ocean unless they were on a ship in the sea, right? So what did they do? They started a campaign, Save the Whales. They started to show videos of the whales, play sounds of the whales. People started to hear this and begin to feel like, wow, really attached to these creatures that they, you know, seemingly have no effect on. Now everybody cares about saving the whales, right? Like, if I'm at the beach, like, I pick up the trash that's on the ground. Is it directly to save a whale? No, but it's because I've become aware of these organisms that are on this planet with us, and I want to do the best for them, right? So this happens too with microbes. People suddenly say, wait a minute, what about my microbes right here? I want to save them. So what's the best way to save these specific microorganisms? And it's going to be to get those native plants. Now, we're not always allowed to go out to these special places. There's wilderness areas here. There's not a lot of them, but there's some. And we're not allowed to just go out and start hacking branches off the trees to make compost. But what we can do is we can keep our eyes on these building projects. You know, when they come in and they just like completely destroy the forest? We need to be up on that. We need to get in before they take those items to the dump. We need those shrubs, those trees, those grasses Elaine's talking about, and we need to make compost out of those. Before the destruction goes to the landfill, somebody's got to intercept it and help those microorganisms that are covering all of those plant surfaces to grow. We went over this yesterday in our class. We're going to do it again tomorrow. And we're going to really focus tomorrow on how to build an inoculum without using something that's store-bought. So we're going to continue to hammer on that and repeat. And you know, I think we need to give Elaine a little bit. I was grateful for your time, Elaine.
Just 1 second here, guys. Thank you so much, everybody. Thank you. We've got some very special stuff here going on, guys.